Welcome to another lecture. This lecture is about secondary metabolism. So when we talk about secondary metabolism, what we are talking about are the types of metabolic reactions that are different from the primary metabolic reactions. And the primary metabolic reactions are the ones that involve sugar production and breakdown, such as glycolysis, gluconeogenesis, the tricyclic carboxylic acid cycle, so the Krebs cycle, fat breakdown like beta oxidation, electron transport chain, and anything that has to do with protein synthesis or amino acid synthesis and breakdown. So those are primary metabolic pathways, and the ones that we're discussing today are secondary ones. And they're particularly interesting in plants because plants have so many cool products that they're making through these secondary metabolic processes. And some of these classes include the terpenes, the phenolics, and the nitrogen-containing compounds, which are then again subclassified as the alkaloids, cyanogenic glucosides, and glucosinolates. And so we'll talk about all of these today, and then try to, in later lectures, try to connect the secondary metabolism with the primary metabolism so that we know where these particular pathways that I will discuss with you today branch off from primary metabolic pathways. But let's take a look here at the terpenes first. So terpenes can be identified because they have usually multimers of C5 compound structures. And this is the C5 compound structure, isopentenyl diphosphate. So isopentenyl has 5 carbons, 1, 2, 3, 4, 5. And it has these 2 phosphates. So isopentenyl, iso because it's not in a straight line, it has an isoform here, so there's a fork, and then it has the 2 phosphates. So it's phosphorylated isoprene, that's also the name for it at some point. So you'll see that all these terpenes have multimers of these C5 structures, and I'll point some out, but it's always a good way to identify a terpene by looking for these multimers of 5s. So some of these terpenes are then classified by the number of multimers of 5. So you have the monoterpenes that have just 10 carbons. You have the diterpenes that have 20 carbons. Triterpenes, tetraterpenes, and then sesquiterpenes are 15 carbon compound containing terpenes. So some of these monoterpenes include anti-herbivory volatiles that are often found in trichomes of leaves. They're called glandular trichomes because they're sort of like little glands that can burst and then release the substance that's inside them. Gibberellins, brasinosteroids, and also abscisic acid or ABA are all plant hormones that will be discussed in more detail later on in this class. And then C40 tetraterpenes are carotenoids. Okay, so let's see how this isopentenyl diphosphate is produced. So you'll see that at the end, here's our end product. That's the isopentenyl diphosphate with the 5 carbons. This is what's then being put together into the terpenes. But how is this product made? Well, that's made by, first of all, starting to put together 2 compounds that are called acetyl-CoA. So acetyl-CoA is an activation compound. It's used for many activation steps in all kinds of pathways. The CoA is a pretty large compound or structure that is stuck onto other parts that activate them. So here all you see is this CoA abbreviation, and it's there's a sulfur bond here, and then you see the 2 carbons. So 2 carbons, that's the acetyl, right? The part of the acetic acid left over. So you take one of those, you take another one of those, you put them together. The CoA is split off, and now you have 1, 2, 3, 4 carbons. So add another acetyl-CoA to that. Here, 2 more carbons, and you end up with 1, 2, 3, 4, 5, 6 carbons. And this product is then reduced by NADPH. So hydrogens are added, and you end up with mevalonic acid. So this is mevalonic acid. Now adding on the phosphate groups so that you get the isopentenyl diphosphate in the end. So one ATP here, another ATP over here, and you're getting to mevalonic acid diphosphate, which then is one carbon dioxide is split off, and that gives you a total here of 5 carbons left, isopentenyl diphosphate. And so then this is our structure that we use for making all these terpenes. So if you look at terpene formation, there is a multitude of ways how you can make all these terpenes. So you can start with isoprene. This is isopentane. So just as an example to see where the double bonds are. So this is what distinguishes the two compounds. And then what this figure is trying to show is that isoprene can be stuck together either in a head to tail formation. So you see basically two of these head to tail, or head to head. So that will give you a slightly different structure, right? If the two head groups are fused, or head to middle. And then you get these really weird structures that look like triangles and that stick up and are sterically very complicated. So sometimes it's more difficult to then distinguish why these were terpenes in the first place. But we'll first start to take a look at some simple terpenes, and then you see some more complicated terpenes and derivatives of terpenes. All right, so here's a nice example, limonene. So if you rub lemons, or you take the peel off from oranges and you sort of squirt them, you see that there's this oily substance that comes out, sticks on your fingers. And you can take this orange peel or lemon peel and you can squirt it in a flame or a candle or something, and you will hear it crackle and flame. So it's flammable stuff that's in there. And that is this limonene, and there's other compounds in there as well. But limonene is one example of a terpene that's found in citrus fruit. And so you see here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 carbons. So 2 times 5, this is a classical monoterpene then. So they all sort of look like these head groups with 2 feet, head, body, and then little antenna like a little green Martian or something. So this is a classical monoterpene structure. And you see that here as well, slightly different with the double bonds. This is menthol, comes from this plant over here. And here's an example for glandular trichomes. So this is a scanning electron microscope image where you have all these trichomes, these little hairs that come out of the leaves, and they have these glands on the top, or fused bodies that have the terpenes inside. And so when they pop open, they will release the terpenes. So here's another set of examples for terpenes. Limonene here at the beginning, and then here you see one that is head to middle fused. So you see that this is now more complicated. You can still count the 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 carbons. So they're still just made up of 10 carbons. Here's one that has 15, so that is an example of a sesquiterpene. That's abscisic acid. That is one of those hormones that I was talking about earlier. Here we have a monoterpene derivative, so now you end up with structures that are looking a little more complicated, a little more different than just these stick figures. So here you have your 1, 2, 3, 4, 5, 6, 7, 8, 9 carbons, right? So there's a derivative. It starts with the 10, and then there's all kinds of changes that occur. This is catnip. So this is an example. You may have heard of catnip. Comes from a mint plant and has this stuff in the glandular trichomes. Cats love it. Here's an example for triterpene that's called brasinolide. So that is a brasinosteroid, a plant hormone that is relatively similar in its structure to steroids that you find in animals. And so it's slightly different though, but that's why it's called brasinosteroids, because they were first found in the Brassicaceae, where they are produced at other plants, other species as well. And here's an example of another derived aromatic terpenoid with a C21 structure. And so that is tetrahydrocannabinol, or THC. And so you see that you can count up the carbons, you find out that it's 21. Here it's difficult to see that this was originally derived from a simple terpene. All right, let's move on to the phenolics. So there are multiple types of phenolics. We have simple phenolics. Some of those are allelopathic in nature. What that means is that plants produce these so that they're toxic to other plants, and so they can mark their territory, if you will. Black walnut is one example where the walnut tree will have basically a sort of space around it where nothing else will grow, because it produces this allelopathic substance, and it's a plant toxic compound. But there are also other examples, anti-herbivory or attractants. So anti-herbivory means plant producing a substance so that it doesn't get eaten by an herbivore, or attractants for pollinators, for example. We will consider many of these as spices, so we are basically attracted to these as well. So they're also human attractants, and we consider them as spices. I have some examples for you here. So here's a phenolic. Phenolics, now phenol, you probably remember from organic chemistry. They have the benzene ring and then have a hydroxyl group. And so then derivatives of that are phenolic. So you'll see these structures with a 6-carbon ring and the hydroxyl group. So that is our phenol. And then you have multiple derivations from that. Here's an example of vanillin, which of course is the major compound of vanilla. This is also the substance that you will buy if you buy vanilla extract, well, not vanilla extract, but the cheap stuff. So vanilla extract will have not only vanillin in it, but all the other substances, other phenolics, other compounds that come from actual vanilla. But if you only produce this will have a pretty good vanilla flavor, and it's much, much cheaper to produce. So vanillin is what you get 100% of when you buy that in a store. Vanilla will have vanillin in it if you buy vanilla extract. So that's the differences here. Here are some examples of phenolics from green tea, or from cloves, nutmeg, cinnamon, from coffee beans. So you see that this is quite a bit of a derivative as well, but you always see the original structure. Here's the 6-member carbon ring with a hydroxyl group, and here the hydroxyl group has been changed so that you have this aldehyde group. So it's cinnamon aldehyde. Here you see the phenolic, and here you barely see the phenolic left. Okay. So we have the simple phenolics, for which I just showed you an example. So next, I'm going to show you some other phenolics that are called coumarins. So this is a subgroup of phenolics. These are anti-herbivory substances that plants make, and they are toxic after being light activated from UV light. And what can happen then is that these coumarins can bind to DNA, and I'll show you a picture. So coumarins are also furanocoumarins. So here furano, this is a furan ring, 1, 2, 3, 4 carbons, and it has this oxygen in there. So that's a classic furan ring, a 5-member ring, and together this is called a coumarin, or a furanocoumarin. So you see the furan ring here, you see the furan ring here. This is this substance is called psoralen. This one is 8-methoxysolarin, and this is a substance that is found in Heraculum. You might have seen this around, these kinds of plants. So some of them, many of them look similar. They have these umbels. Umbelliferae is the plant species, or the family, and this is the substance. And when that substance is activated and by UV light and binds to DNA, it can lead to this nasty blistering. So here you see the blisters on the hand. So that's what happens when it gets sensitized. Psoralen, interestingly, is also a substance that has been used for marking DNA in the molecular lab. So molecular biologists would use this, put a little label on it, something to make it maybe so you can bind it and then isolate it, or so that you can visualize it, and then just mix that with DNA, hold it to a UV light, and it will bind to DNA. So it's an easy way to mark or to label DNA. So that's the psoralen. Okay, so we had the simple phenolics, we had the coumarins, and now flavonoids are also phenolics. So you see that the structure becomes more and more complicated, less simple. It's no longer a simple phenolic, but it is made in the plant by steps that use simple phenolics as starting material. So what do I have down here? This is these are all flavonoids, so they're all similar in structure. You see that they all have these 3 rings, and then they are stuck together with this connection here. But there are differences, right? Differences with respect to how many hydroxyl groups, where the hydroxyl groups are, where this third 6-member carbon ring is located, whether it's pointing up or pointing down, if you will. And so that is what gives them the different names, so the different subcategories of flavones or isoflavones or flavonols or anthocyanins, and then there's several more. These are the 4 that we'll talk a little bit about. What you can see is that they all have this relatively similar structure in general, and so all of these are flavonoids. So the anthocyanins, as a particular type of flavonoids, all are colored. Many of them are colored, and you can see them here as examples of the chemical structure. And here you see essentially the color that they produce in the plant. So this is larkspur, rose, and geranium. And so this makes this nice red color, this makes sort of a pinkish color, and this makes the dark blue color. So all of these have this C6, C3, C6 structure. So C6, 1, 2, 3, 4, 5, 6. And then you see there's an oxygen here, right? So 1, 2, 3 carbons only. And then here's a 1, 2, 3, 4, 5, 6 carbons again. and that makes up the anthocyanins. And then you see the difference is only the hydroxyl group location. So you have 1 here, you have 2 here, you have 3 here. And that changes the color, the color as we see it. Bees see it also. Bees see it in a different color on petals, for example. But this is the only thing that makes the difference between the electron organization within this compound and what types of light wavelengths they can absorb, reflect, and therefore produce a certain type of color. Okay, so we talked about the flavonoids, and I showed you some examples of anthocyanins. Flavones and flavonols are oftentimes bee-attracting colors, so colors that bees can see. Sometimes humans can see them, but they might be in a spectrum that is slightly outside what we consider the visible spectrum. They also work as UV screens for plants. So remember that UV is also, it's not just bad for human skin, it's also bad for anything that has DNA, because UV will mutate DNA. And so having a substance that covers the DNA, or that covers the outside of the cells, of the tissue really, will help with against mutation, and is used as attractant for rhizobacteria. And then the flavonoids are, so those include some insecticides, some pesticides, antibacterials, anti-estrogenic substances. So those are substances that reduce the estrogen response. And some of them have anti-cancer properties, which you may have heard of through the popular press. So flavonoids and flavones, anthocyanins, reactive oxygen scavengers, anti-cancer properties. Those are sometimes things that you hear about because they get discussed as supplements. And then there are complex branched phenolics. So all of these are phenolics. Remember, all of these are just examples of phenolics, and this is another subclass of phenolics. This is not a flavonoid. This is a complex branched phenolic. Let me show you one of those. Lignin, you probably know that lignin is what makes woody plants woody. So that's the stuff that makes a tree really strong. It's not just the cellulose. The cellulose makes up most of the cell wall, of course, and you will find that in wood products. But when you hear the term wood-free, like wood-free paper, for example, what that means is that the lignin was removed. The cellulose was kept. So here's an example of a branched phenolic, and you see always these rings, and you see hydroxy or oxygens that at some points were hydroxyl groups, and then were derived or used for making the derivatives of these multiple connections between all of these substances. So they can be very complex in their branching, and this type of phenolic is lignin. So there's not just one type of lignin. Lignins are look like this, and they can be hundreds of molecules long or big, and they can be branched in all different kinds of ways. You see these little squiggly lines. That means that the molecule isn't over there. Okay, so those are some of the phenolics. Now let's look at how these phenolics are made, right? We wanted to see where the metabolic pathways branch off, and then later on in the semester we will discuss how the secondary substances are connected to the primary metabolites. But in order to do that, we need to look at some major pathways of these secondary metabolites. And so one important one is the shikimic acid pathway. I write here the most important secondary compound pathway for phenolics. And then there's the malonic acid pathway, which is more prominent in bacteria and fungi. Well, let me show you the shikimic acid pathway. We'll come back to that at some point. There are some interesting parts of that pathway. I'll point a few out as I walk you through this. Look at the end first. So from this pathway that goes like this, we will have end products such as alkaloids, phenolics, including the flavonoids and coumarins. And all that is possible because we're producing, through this shikimic acid pathway, aromatic amino acids. Right? So these aromatic amino acids are the ones that then give rise to the alkaloids and the phenolics and so forth. All right, let's see how it's made. So it starts with a erythrose 4-phosphate. You haven't really seen that substance yet. I think it might have been on one of the pathways that we looked at so far. We'll come back to that. This comes from the pentose phosphate pathway. We briefly discussed the pentose phosphate pathway before in class. So the erythrose 4-phosphate, 1, 2, 3, 4 carbons, and then a phosphate group. And then you add phosphoenolpyruvate or pyruvic acid here, so depending on the pH. This comes from glycolysis, right? So from almost at the end of the glycolysis pathway, you have seen this before. So this plus that will split off the phosphates and will produce shikimic acid. So this is shikimic acid here, this substance. 1, 2, 3, 4, 5, 6. 3, 4, 5, 6, 7 carbons. And then it has the 3 hydroxyl groups here. So add an ATP to activate it, and add another phosphoenolpyruvic acid, so PEP, phosphoenolpyruvate, and you get to this substance here, 3-enolpyruvyl shikimic acid 5-phosphate, or EPSP. This will then produce chorismic acid, and chorismic acid is further metabolized to aromatic amino acids. Why am I harping on these substances here? Well, this is a substance that is catalyzed, so this reaction is catalyzed by EPSP synthase, or EPSPS. This substance here, this enzyme, is the enzyme that is vulnerable to herbicides. And so herbicide resistance, therefore, can play with this EPSP synthase in order to make a plant herbicide resistant. And then if you have a plant that is herbicide resistant, because this enzyme here has changed in its structure slightly so that this herbicide doesn't affect it anymore, can then produce resistance. And the herbicide that I'm talking about is Roundup. So the Roundup Ready crops, the first Roundup Ready crops that were made, had a mutation in the EPSP synthase gene. And so therefore, when you put Roundup on the plant that's sensitive, then this enzyme will be blocked. This will not be produced here. This pathway is blocked, and no aromatic acids are made. Not even, thinking about all the secondary compounds that are not being made, but also the aromatic acids are not being made. So that is how Roundup then kills plants, and how Roundup-resistant plants are made stable, or how their EPSP synthase is made stable. We'll talk more about that, but this is the pathway that we will come back to at this particular step then. So I just wrote down here a few things to remember. So the shikimic acid pathway made from phosphoenolpyruvate and erythrose 4-phosphate. The EPSP is catalyzed by EPSPS. That enzyme is inactivated by glyphosate, which is the common name, or the store name is Roundup. And then Roundup Ready crops contain this glyphosate-tolerant EPSP synthase. Originally found from bacteria, but there are now many different forms that have been made basically through directed evolution. So you can, dump as much Roundup on a plant, basically waiting for mutations. You can do a mutant screen, and then you can find enzymes that are resistant, enzyme forms of this particular gene that are resistant to glyphosate. All right, and so now let's get to the nitrogen-containing compounds. Among the nitrogen-containing compounds, there are three classes that we'll discuss. The alkaloids are probably the most interesting, or the most notorious, if you will. And then there are the cyanogenic glycosides, cyanogenic, so they're producing cyanide, and glucosinolates. I'll show you those in a little bit at the end as well. So all of these have in common that they contain nitrogen. That's really the only thing. The compounds look very different from each other, but they all contain nitrogen. So the only thing that gives them a particular structure that is then lumped together is this group number 3, is the nitrogen. Okay, so alkaloids the group of alkaloids contain anti-herbivory plants, anti-herbivory compounds found in plants. They're toxic to mammals. Sometimes they are toxic to livestock. So there are plants that farmers want to make sure do not grow in their fields because they can be toxic to livestock. There's many notorious poisons that are in this group. Strychnine, for example, coneine from the hemlock plant. And in small doses, some of these alkaloids can have medicinal properties. They can act as stimulants or sedatives. And I'll show you some examples. So here are some chemical structures of all these alkaloids. You see that they contain nitrogen. All the nitrogens are highlighted here in red. So here's cocaine. That's from this plant right here. And then we have caffeine, which is from this plant right here, so coffee plant. So you see the caffeine molecule has multiple nitrogens. Atropine is a very interesting compound. So that comes from Hyoscyamus, belladonna. So we'll talk a little bit more about that in a second. And then you see heroin here. This is quite complicated, right, with these bridging carbons. Here's the nitrogen, and nicotine, Conine, that's this poison from the hemlock plant that I mentioned earlier. And so I just wanted to show you the heroine right here. So you see this substance that has, you know, the two 6-membered rings, this one, and then here's one more, and then has this oxygen group here, and then this weird structure with this bridge, and then derivative groups up here. So if you look at this is codeine and this is morphine. Those are all very similar. They are derived from poppies, and you see that this part here is different. This part here is different, and that's the part that's different from the heroine as well. But you see that many of the other parts of the substance are the same. So very similar chemical structures with slight variations in the outcome here. And here's an example from poisons, alkaloids that you find in leaves of potatoes. So that's why you don't eat the green parts of potatoes, right? When you keep the potatoes in the drawer in the dark so that they don't start to green up in the green stuff once it reaches light, once it deethylates, and then starts to produce secondary compounds in its leaves. That's when you don't want that to be transported into the roots, or if you were to take the eyes of the potato that are basically leaves that are young leaves that are being grown out, they can start to produce these solenides, solanine, that are nitrogen containing and poisonous. Okay, so I want to talk about two specific alkaloids and their effect on humans. It's just such an interesting topic that even though this is a plant physiology class, let's take that little detour here. So if you look at the neuron that is transmitting information from a receptor to an organ that responds to the neurological input, and you consider the effect that cocaine has on this, so we just talked about that as being an alkaloid, right? So cocaine will interfere with the transporters of these neurotransmitters. So neurotransmitters are little chemical structures or little chemical compounds that are produced and that basically transmit the electric signal from one neuron to the next neuron through the synaptic cleft. So there's a space here between these cells, and in order for this neuron to talk to that neuron, a chemical response is used in the little space in between. And so that chemical, one of those chemicals, neurotransmitters, is dopamine, gets packaged into these little vesicles, gets then excreted from the cell through these vesicles into the cleft, and then taken up again from uptake transporters. So you have the dopamine being released, and then what it's doing is it's looking for the dopamine receptors on the other side, on the receiving neuron, and so that is producing the stimulus, and then at some point it has to go away, right? Otherwise you would have constant stimulation. And so in order for that, like a telephone, right? If the telephone rings all the time, then you'll never pick it up because it's no longer serving a function. So you want that signal to be received, and then you want the signal to go away, and the signal goes away by this uptake transporter. So what does cocaine do? Cocaine binds to the uptake transporter, and so it basically blocks it. And that means that dopamine increases in its concentration in this synaptic cleft, and it kind of bounces around, and every time that it hits one of these receptors, it adds another signal. It adds another transmission of response. And even if there isn't anything that that should potentially be noticed, this is what increases this euphoria, or this extra energy, as it's sometimes described, because the uptake, the signal cannot be turned off. So the telephone rings the whole time, basically. So that is what cocaine does. And then at some point it gets broken down, opens up the channel again, and that cocaine high dissipates. So I wrote a few things on here. Effects of cocaine. Dopamine reuptake is blocked. The dopamine continues to stimulate the receiving neuron. That's the stimulant effect. There's an interesting YouTube video that I found. You might want to watch that. I found it interesting. It's 4 minutes long or so. Okay, and the second compound I wanted to spend just a few minutes thinking about, and that is atropine. So atropine comes from the plant henbane. Belladonna is a term that's been used. So this substance dropped into an eye will open the pupils, and so people actually put that stuff in their eyes hundreds of years ago, maybe not even that long, and in order for your eyes to dilate and apparently appear more attractive or prettier. And so that's why it's called belladonna. Belladonna in Italian, it means pretty lady. So the substance has that effect, but it has many other effects too. So it's also has these neurological effects. And so here you have another example of a neuron that is interacting with a receiving neuron. And so acetylcholine is the neuron, the neurotransmitter here. And you have the transmission, the binding to the receptor, and the uptake. And so what's happening in this case is that when you have atropine in the system, the atropine binds to the cholinergic receptor, so the choline receptor or cholinergic receptor, and it blocks the transmission. So now you don't have this extra euphoria, you don't have this extra stimulation, you have the opposite. You have this, everything slows down because the signal cannot be transmitted anymore. So it distorts perception, and then over time the block will be, metabolized, will be broken apart. But there are antidotes, and there's a substance that's called galantamine that is found in daffodils that binds and then activates the acetylcholine esterase, which normally breaks acetylcholine down and helps the uptake. And when that is not broken down, then that increases the concentration of the acetylcholine in the synaptic cleft. And because of chemical equilibria, essentially, the more you have of this substance, the more likely it is to displace the other substance that binds to the receptor. So you increase this concentration here, and therefore the competition gets stronger, and so you basically wash away the atropine. So that is how the antidote works for the atropine, and it's been sold or discussed, mentioned as a potential anti-Alzheimer medication. I don't really know how far along that is, whether that is a real thing or not. But it's an interesting, historically interesting compound from plants, because if you think about these substances, all of these substances that come from flowers that you grind up, there's a daffodil and there's this henbane. Those are the substances on which the stories are based, that have to do with witches and witch trials, and people thinking that botanists, essentially early botanists, that ground up plants to extract certain substances from them and then tried them out on themselves, had these effects, and they could be either toxic, or they could be stimulating, or they could be drug that induces some sort of a changed perception. And so you can easily see how, especially this substance, and especially this response where neuroperception is slowed down, those are supposedly the substances that were used when witches would describe afterwards what they saw, what they felt, that had to do with these witches' rides on broomsticks flying through the night and so forth. So apparently that is the kind of change in perception that then induced these dreamlike states where stories then came up from that. All right, so here we have two alkaloids with interesting history. Okay, let's spend just a few more minutes on the nitrogen-containing compounds, class B and C. So those are the cyanogenic glycosides. There's one of those, and then we have the glucosinolates. But let's look at the cyanogenic glycosides. They release toxic HCN, so cyanide, when the plant is injured, and that is because the substance that has to be broken down is stored in one part of the plant, and then another part of the plant, a vacuole, for example, will contain the enzyme that will break down this glycoside. And so only when the cell is actually physically broken and the vacuole injured, popped, then these two substances come together in contact, and then the following reaction occurs. So here you have your cyanogenic glycoside, not toxic, and the glycoside, not toxic by itself, but when they come together, the sugar is split off, which detoxifies this compound. And through these intermediate steps, you end up with cyanide, hydrogen cyanide, right? So this is extremely toxic, and so any herbivore that chews on leaves that have these cyanogenic glycosides in them will create this super toxic compound, and that is not only, well, it's probably deterring future feeding when you're dead, but that's certainly a problem for livestock that encounter plants that have these substances in them. I would assume that very small quantities of this might deter feeding, but this is a very toxic substance. A not quite so toxic substance are the glucosinolates. They come from the Brassicaceae, so rapeseed, for example, canola, those are the kinds of plants that are in the Brassicaceae, mustards. So releases a somewhat toxic isothiocyanide. It's kind of bitter as well, so it's maybe not so toxic. Maybe it gives the animal a stomach ache, but won't outright kill it. Same thing happens here. The glycoside and the enzyme, they break down the glycoside when they come together, when the animal starts chewing on the plant. And then it probably stops pretty quickly, moves on and eats something else. And here's the reaction for that. So you have the glucosinolate bound to glucose. The glucose detoxifies this substance. Then you have the reactions that break off the glucose, so it's the glucosidase. And then you have the isothiocyanate that is produced, and that is the substance then that causes the anti-herbivory effect. So that is our third subclass of the nitrogen-containing compounds. And with that, that's all for today. I will see you in class. One more thing. So you might ask, what do I need to know about all these substances? Do I need to learn any chemical structures, yes or no? And the answer is as nebulous as my answers have been in the past. It's great to know structures by sight. So it's great to be able to see a structure and say, oh, that is a glycosinolate, or this is a monoterpene, or this is a diterpene, or this is an anthocyanine. I would never ask you to write down the chemical structure, of glucosinolate, but I would hope that you would be able to look at a structure let's say like this, and see that it has, is derived from many phenolic compounds, so that it's a phenolic. So, and that you could see here that this must be an anthocyanin because it has this C6, C3, C6 structure. So if I gave you something like that and said, well, what kind of structure is that? Then you should be able to say, well, it's an anthocyanin, or at the very least, you should be able to determine that this is a phenolic of some sort. Same thing here with the terpenes, right? So if I gave you something like that, that should be really easy enough to just count to 10 and say, oh, there must be a monoterpene. I wouldn't give you something like that and then say, well, what is that? Because this looks like could be a phenolic here, but it isn't. So that's not something you can simply see from the structure. But if you could look at these structures enough so that you can tell, what they generally are, and then when it comes to the pathways, we will want to talk more about the shikimic acid pathway. And so it would be good to look at that a little bit. Again, never you have to write down any of these structures from memory, but you should know, you should be able to recognize amino acids, especially aromatic amino acids. You should be able to recognize, counting carbons and sort of seeing where they come from in primary metabolism. Now, we'll talk a little bit more about that in the upcoming lectures, so no need to just memorize all of these at this point, but do start looking at your structures and do start to have an idea of what they are, what kind of class they belong to. All right, see you in class.